Hello class, good morning. I can see that there are only two students have joined us, but mostly it's 30 students. So in that case, I cannot start my class. This will be not fair. Um, I think no one is interested, I can say. Here we can see concurrent viewers like three. So in that case, until and unless I get two-digit figure, I can start the class. Otherwise, we start at 7, normal 7 p.m. then. But you all three are present students. Ankur, Tiwari ji, Shubham, you all three are present. Just confirm me who are present by saying yes. Based on that, we can start our class. Okay, Ankur is there. But it will not make sense to start the class. Shubham is there. Because you wake up for me, that's good. I appreciate that. I also wake up for all of you early. But thing is, um, we cannot start with a class with two or three students. Otherwise, we continue at 7. Okay, 5 is there. Okay, let's start. Let's see. Protein isolation, quantification, and purification. So yesterday there was buffering was happening, so we couldn't discuss much. So these are the famous people who, the Nobel Prize winners, who got the in the field of chromatography and electrophorese for the partition chromatography, basically. Your Martin, Singe, Tislas. Then comes your Fan, Tanaka, Swedberg. They got on the various fields. Tanaka got in mass spectrometry. Fan got the Nobel Prize for the biological micromolecules, whereas Swedberg for the dispersed system. And Claude, Douay, Plade, they got for the organelle fractionations of your cells. Then this is Frederick Sanger, who got 2 times Nobel Prize winner, one for the structure of insulin and another for determinations of sequences in the nucleic acids in 1980. How you can isolate protein? For isolation of protein, you need to first disrupt your cells, yeah. Then crude separations of homogenates into the fraction. Then homogenization is done with the help of Downs homogenizer, sonication, osmotic shock, and French press. Then fractionation has to be done with ammonium sulfate precipitations, differential centrifugation. So what happens in centrifugation? You have homogenoids. You centrifuge for 500 G for 10 minutes. Then in the pellet you got the nuclear fraction. Then after 10,000 G for 20 minutes, in the pellet you got mitochondrial fractionation. Then in the supernatant you got 100,000 G, and that's contains cytosol, and it's a microsomal fractionation in the pellet. In this way you can separate between different molecules. And there's also two types of centrifugation: differential and rate zonal. In the differential, you mix together your dense and less dense particles, more dense and less, and then you go for centrifugation. In the pellet, you get the more dense particles, and the less dense particles remain in the supernatant. On another case, you have the sucrose gradient. In the sucrose gradient, you have a mixture of your large and small particles. Then you go for centrifugation, so they get differentiated according to their density. So thicker will be bottom and less dense will be upper. You can separate them separately. So at different speeds for 3,000, 10,000, 40,000, 200,000, 400,000. You got different proteins results. Then how you do the protein quantitation? There are two methods like Lowry Bradford. Lowry is the quite old one, which could determine the protein concentration from 10 microgram to 1000 microgram per ml. Whereas the Bradford, which is simpler and faster than the Lowry method, and in this we use the Commercy Blue G250 dye, which appears to be most readily available and get attached to the arginine and lysine residues of your proteins, and they could give you about 5 to 25 microgram per ml. So let's check how this thing is done. Yeah.
In this video, you will learn how to perform protein quantitation using the Bradford assay. Protein quantitation is one of the most commonly performed procedures in a biotechnology laboratory. In this activity, the protein concentration in milk will be measured using the Bradford assay. For this activity, you will need 1x Bradford reagent and 1x phosphate buffered saline, or PBS. Using a 20 to 200 microliter micropipette, set the pipette to 98 microliters. Pipette 98 microliters of 1x PBS into an empty microcentrifuge tube labeled sample. Switch to a 2 to 20 microliter micropipette and set the pipette to 2 microliters. Pipette 2 microliters of milk sample into the microtube labeled Sample A. Mix the milk sample and PBS by either pipetting or vortexing. This mixture has diluted the milk to 1 part in 50. Label 2 cuvettes, one control and the other sample. Do not handle the cuvette where the light passes through, and make certain your labels are well above this area. Pipette 20 microliters of diluted milk sample into the cuvette labeled sample, and 20 microliters of 1x PBS into the cuvette labeled control. Next, we will set up protein standards. See your protocol for the correct concentrations. Label a cuvette for each standard and mark it with the corresponding concentration. Next, add 20 microliters of each protein standard into the corresponding cuvette. Make certain you use a clean pipette tip for each sample. Now you are ready to add the Bradford reagent to all of the cuvettes you have prepared, including the sample and control. Add 1 milliliter of 1x Bradford reagent to each of the cuvettes. Mix completely by pipetting up and down with the micropipette. Incubate the cuvettes at room temperature for 5 minutes. After 5 minutes, visually compare the cuvette containing the milk sample to the cuvettes containing the protein standards. Determine the standard that most closely matches the color of the milk sample, estimate the protein concentration of the milk sample based upon the visual comparison. To determine the protein concentration using a spectrophotometer, select for the Bradford assay. When asked to insert blank into the spectrophotometer, insert the cuvette labeled Control. Use the blank to set the spectrophotometer to 0 absorbance, or 100% transmittance. If instructed, read the absorbance of the 7 protein standards and record the absorbance values. Remove the cuvette labeled blank and insert the milk sample into the spectrophotometer. Read the absorbance and record the value.
So that was the basics about Bradford assay. You can also do um, another technique which was published in Biotechniques paper, which names ultra-sensitive fluorescence-based protein quantitation, in which you add a dye to your samples, then heat for 10 minutes, and cool to the room temperature, then you can read the fluorescence. So in this way, you have one on-site fluorescence, another side protein. So once you get the fluorescence, you can predict the protein concentration. Then we are done with the isolation of a protein. Now it's time for to quantify, to purify your protein, how you can purify the protein. So in order to do so, for protein purification, you have your dialysis bag, you have concentrated solution, and you have buffer outside. Yeah. So you put this into the dialysis bag at the start of dialysis, and then at equilibrium, everything starts to get differentiated, and you will have mainly the large particles inside of your bag. So let's check. Yeah. Then there's some other way, like with the help of chromatography, you can differentiate between your proteins. First example is your by gel filtration chromatography, gel exclusion chromatography, we also call that. So you have a protein sample, right? And then you have a gel. So your particles are yellow, green, and red. The red particles are the smallest, green are the middle, and the yellows are the largest. And you have your carbohydrate beads. Yeah. So what will happen? The smaller particles will stay inside these beads. The green and the yellow one they will go transfer. So according to that, the differentiation will happen. So we can see here also in the same example. So let's check how this gel filtration technique is done. Cefadex G50.
We're starting with gel filtration medium. This is Cefadex G50. The number here gives you an approximation of the size exclusion. So this is will exclude proteins above 50 kilodaltons, 50,000 molecular weight. Inside is a white powder. In the speaker I put 9 grams of the G50 powder, and I swelled the powder overnight using this tris buffer, 30 millimolar tris buffer, pH 8.0. You can see that the 9 grams has swollen up to a volume of about 90 milliliters, almost 10 milliliters per gram of solid. Now I'm going to gently stir the gel. It's important not to stir this too vigorously. Don't mechanically stir this. That can break your gel particles up into very fine dust, which will impede your flow rate in the column. So this is making a slurry. I'm using a sawed-off pipette to transfer my slurry to the column. For gel filtration, I want a fairly tall gel bed. So I'm trying to mostly fill this column. And excess buffer will drip out the bottom. So here you can see the drips coming out the bottom. Gel bed is starting to settle a little bit. Gel bed is right here. I'll add a little more buffer so that it doesn't run dry at the top. So I'm gently adding a little buffer to the top. This looks great. You can see the gel bed is fully settled here. There's a little bit of buffer on the top. I've capped the bottom right now to keep it from going dry. I'm going to put a mixture of 2 molecules onto the column. Blue dextran is a very large molecule. It's a polysaccharide. Molecular weight about 1 million. Fluorescein, on the other hand, is a small molecule. Molecular weight about 350 grams per mole. I chose these 2 molecules because they're nicely colored, so I'll be easily able to see the separation visually as they go through the gel filtration. I've drained the buffer, so now there's no residual buffer on top of the gel, and I've made a mixture of a total of 0.8 milliliters, 400 microliters of the fluorescein, 400 microliters of the blue dextran. Nice green color. I'll separate these on my gel. For this size column, this is getting near the maximum I would put on the column. I'm using a Pasteur pipette to gently load my material onto the column without disturbing the gel bed. So here you can see the blue molecules, the large molecules, are moving faster through the gel. So I'll hope that I can get a complete separation between the blue and the yellow. Now the excess on the top of the gel is all soaked in. So I'm adding buffer very carefully to the top of my gel filtration column. I'll just add a fraction of a milliliter first to help get everything into the gel before I add a large volume of buffer. So now the separation is proceeded somewhat. I'm going to try to add a decently large volume this time of buffer on the top. About a milliliter would be great. If I do this well, then the buffer that I put on the top remains colorless because all the colored molecules have already gone into the gel. Alright, now you can start to see a little colorless layer on the top. That's perfect. All the colored molecules are in the G50 and they're starting to separate quite nicely. the yellow molecules are up here, the blue are down here. Still got a little overlap. So I may have overloaded this column, but we'll see. I'm hoping that before the blue molecules elute that they'll actually be separated from the yellow, but it's going to be close. So out of the bottom now, blue liquid is dripping. You can see I do have a colorless layer starting to develop here. The fluorescein has been slowed down by the gel filtration. The blue dextran is coming through. So this technique is often used to separate a protein, which is large, from the small molecules that could be salts or could be a fluorescent tag that is unwanted. I've topped up with buffer on the top so that it never runs dry. The fluorescein is now eluting from the column. Fluorescein is fluorescent. I'm shining a little blue light, UV light on it, and you can see it glows green. The last little bits of fluorescein are emerging from the column now. Here's the fractionation. This first tube has nothing in it. So this volume, which looks to me about 2.5 milliliters, is called a void volume, where nothing comes out. But then the blue molecules, the blue dextran, came through the column swiftly. So this is about a milliliter. Some of the blue dextran tailed into the next fraction. This is low concentration. Then I have a nearly colorless fraction here, and then the fluorescein starts coming out here. Fluorescein stopped coming out right about here. So the total volume will be 2.5, 3.5, 4.5, 5, 6, 7, 8, 9, about 10 milliliters is the total volume, which is what I would expect for a column of this size. Often what you want is just this first fraction with the large molecules. If you're trying to desalt a protein or remove the unbound label from a protein, then this is the fraction that has value. These fractions would just be junk. The original mixture, you can see, glows nicely. And what I believe is isolated blue dextran from the column has no glow in the UV. That's a sign that I have successfully separated out all the fluorescein from the blue dextran. So the column, if I've done it well, comes out completely colorless. It's reusable. Sometimes microorganisms like to grow in the cephadex, so if you want to preserve your column for a long period, keep it in the fridge, certainly. And sometimes azide is added to the buffer to prevent growth of microorganisms. Here's tris buffer with 0.01% sodium azide, and this will prevent microorganisms from chewing on the dextran that's in the column if you're storing it over a long period of time.
Do the right thing. I hope this gel filtration chromatography is understood very well now. So let's continue with the next one. That is ion exchange chromatography. It's the same. We are, instead of having these carbohydrate beads, in this case we are having an negatively charged protein will flow, and we have negatively charged beads. Yeah. So to negatively charged beads, so positive charged beads will be attached, and the negative beads will flow away. So it is of two types: ion exchange and cation exchange. An ion exchange, the matrix carries a net positive charge, which interacts with the negatively charged protein strongly. In the cation exchange, the matrix is negatively charged and interacts with the positively charged proteins. So let's check how it is done.
Ion exchange chromatography is a chromatography process that allows the separation of ions and polar molecules based on their charge. As well as other forms of chromatography, in ion chromatography, there is a mobile phase, which generally consists of buffer solution. And there is a stationary phase which consists of a matrix that contains charged ionizable functional groups. This type of chromatography is subdivided into an anion exchange chromatography and cation exchange chromatography. In anion exchange chromatography, negatively charged molecules are attracted to a positively charged solid support. which is usually beads, that provide a positively charged surface, such as resin quaternary ammonium. In cation exchange chromatography, positively charged molecules are attracted to negatively charged solid support, which is commonly beads, that provide negatively charged functional groups, such as resin methyl sulfate. Ion exchange chromatography is frequently used for the separation and purification of proteins. The basic process of this chromatography can be represented in 4 steps: conditioning or equilibration of the stationary phase, sample loading, washing, and elution of the retained molecules. The first step is the equilibration of the stationary phase to the desired start conditions. The pH and ionic strength of the sample buffer are selected to ensure that, when sample is loaded, proteins of interest bind to the medium, and as many impurities as possible do not bind. Therefore, the same buffer is used for the equilibration of the stationary phase. When equilibrium is reached, all stationary phase charged groups are bound with exchangeable counterions, such as chloride or sodium. When the column is equilibrated, the second step is sample application. Depending on the pH of their environment, proteins may carry a net positive charge, a net negative charge, or no charge. This net charge can be determined by comparing the pHi of each protein with the pH of the buffer. Proteins are large biomolecules, consisting of one or more long chains of amino acid residues. Amino acids are organic compounds that contain amine and carboxyl functional groups, along with a side chain specific to each amino acid. Depending on the pH of their environment, the amino group can be protonated and become positively charged. The carboxyl group can be deprotonated and become negatively charged. And the side chain of some amino acids can become positively or negatively charged due to the gain or loss of protons. So, the net charge on the molecule can be positive, neutral, or negative. The isoelectric point, or pHi, is the pH at which a protein carries neutral charge. At a pH below their isoelectric point, proteins carry a net positive charge. And at a pH above their isoelectric point, they carry a net negative charge. In this example, the pH of the buffer is equal 5, consequently, protein A has a net negative charge. And both, protein B and C, carry a net positive charge. In addition, protein C is more positively charged, since its pHi is higher than the pHi of protein B. If we apply cation exchange chromatography, protein that has a net positive charge, it will bind to the negative charge beads of the stationary phase. On the other hand, uncharged proteins, or those with a net negative charge, would not bind. They pass through the column at the same speed as the flow of buffer. When all the sample has been loaded, the next step is column washing. The column is washed with start buffer. To ensure that all non-binding proteins have passed through the column. When all the sample has been loaded and the column washed, conditions are altered in order to elute the bound proteins. Most frequently, proteins are eluted by increasing the ionic strength of the buffer, or, occasionally, by changing the pH. As ionic strength increases, the salt ions compete with the bound components for charges on the surface of the medium, and one or more of the bound species begin to elute and move down the column. The proteins with the lowest net charge at the selected pH will be the first ones eluted from the column, as ionic strength increases. Similarly, the proteins with the highest charge will be most strongly retained, and will be eluted last. Protein B has the lowest net positive charge, so it can be eluted first. Then, protein C with the highest net positive charge, it can be eluted next, by increasing the salt concentration. Also, varying the pH of the buffer can be used to affect the separation and to elute the retained proteins. As we seen, the pHi of the protein B is equal 7, and the pHi of protein C is equal 9. So if we add a buffer with pH above the isoelectric point of protein B and below the isoelectric point of protein C, the protein B will become negatively charged and can then be eluted. The protein C is still charged positively, consequently, still retained. Next, protein C can be eluted if we apply a buffer with pH above its isoelectric point. So, it becomes negatively charged, and it will pass through the column. One of the primary advantages for the use of ion chromatography is only one interaction involved during the separation. Another advantage of ion exchange is the predictability of elution patterns. By controlling changes in ionic strength or in pH of the buffer, using different forms of gradient, proteins are eluted differently in a purified concentrated form.
Okay. So now next is affinity chromatography. It's basically based on the antibodies available. So antibodies must be available that have been raised against enzyme. So in this we can see there's a glucose binding protein attaches to glucose on beads, and then the glucose binding proteins are released on the addition of glucose. So here we can see we have loaded our sample, the red and blue particles. The red ones are the proteins that could be recognized by the antibody, where the blue ones are the proteins not recognized by the antibody. So when we wash them, so the blue ones will be washed first, and the red one could be washed later. So some drawbacks are that the binding to the ligand or antibody can often be strong, that very harsh conditions are needed to elute the enzyme, and this leads to denaturation. Non-specific bindings may also occur, especially where large ligands are such as antibodies are employed. So let's check how this technique is done. I hope I have some students to be joined with me here today. So yeah, I think the 3-4 students who have started in the morning, they are still with me. The rest students who were not able to come because of the timings, so please go through this recorded versions, and yeah, but we also will have class today at 7 p.m. also, because thing is I am leaving to Kanpur now because our college has been started from Tuesday, and there in Kanpur I don't have Wi-Fi access or internet access, so I have to play with my mobile only. So I want that before I go to Kanpur, I want to done with the maximum part with you. So the rest, if I don't have internet there, I will try somewhere to manage to do it also. So that's why I'm trying it like this today. So let's check how we do this technique.
Separation in affinity chromatography depends upon the reversible adsorption of biomolecules through biospecific interactions on the ligand. Most affinity chromatography experiments are performed in 3 main stages: equilibration, sample application and wash, and elution. The first step is the equilibration of the stationary phase to the desired start conditions. The second step is sample application and wash. The goal in this step is to bind the target molecules and wash out all unbound material. Non-binding molecules will pass through in the flow-through during washing with binding buffer. In the third step, elution, biomolecules are released from the biospecific ligand into the elution buffer by change in the buffer composition. A common way is to increase pH of the buffer.
It's quite simple. Now comes your HPLC, which is a high pressure liquid chromatography, which has enhanced column methods for protein separations. Resin sizes are quite refined in them. Now all the chromatographic methods described above can be applied with HPLC and equipment, but with HPLC separations of the peak is almost always better than the standard chromatography columns. The reason for this is that HPLC columns are packed with much smaller beads than those used in standard columns. So the HPLC, it's a large tube containing small particles known as stationary phase, and the chromatographic bed is composed by the gel beads along with the inside of the column. And the sample is introduced into the injector, then carried into the columns by following solvent. So, we got absorbance at 220 nanometer. So these peaks that we are seeing is 1, 2, 3, 4, 5. They corresponds to different materials, different particles that are present in our sample. So we are able to separate them according to your HPLC principle. So let's check how this technique is done.
HPLC stands for High Performance Liquid Chromatography, but could equally well stand for High Pressure Liquid Chromatography. It is used for separating mixtures, either to analyze the mixture or to separate a required product from others in a reaction mixture. It can also be used to find the relative amounts of different components in a mixture. HPLC works on the same principle as paper chromatography, here shown speeded up. A liquid, called the mobile phase, moves past a solid, the stationary phase. In paper chromatography, this stationary phase consists of water molecules bound to the cellulose in the paper. The mobile phase carries different components of a mixture, called the sample, along with it at different rates. How fast each one moves depends on its relative affinity for the mobile and the stationary phases. For example, if the mobile phase is more polar than the stationary phase, the more polar components of a mixture will tend to move more quickly than the less polar ones. In HPLC, the stationary phase is a solid packed into a column, like one of these. This particular column contains silica particles to which C8 hydrocarbons are attached, making the stationary phase non-polar. In paper chromatography, the solvent moves along the paper by capillary reaction. In HPLC, the liquid is forced through the column by high-pressure pumps. The whole apparatus looks like this. These bottles contain solvents. Two solvents can be mixed in any proportions to give a mixture, the liquid phase, of suitable polarity for the separation that is being done. In this case, one solvent is water, very polar, and the other ethane nitrile, less polar. The operator can decide on a mixture with the correct polarity for the separation she is doing. These are the pumps. They produce a pressure of 15,000 kilopascals, 150 times that of the atmosphere, hence the name high pressure liquid chromatography. If a single sample is to be run, it is injected into the solvent stream here, in the injection port, via a hypodermic syringe. Alternatively, several samples can be run in succession by loading them into this auto-sampler, which will run them in order without any human intervention. The pumps force the mixed solvents through the column. The solvent emerging from the column and carrying the separated components of the mixture passes into the detector. Here, a beam of ultraviolet light shines through it. This light is set at a wavelength that is absorbed by all the components to be separated. When the detector reading drops, the component that is absorbing UV light is coming out of the column and passing through the detector. Many alternative types of detector are possible. This one measures refractive index. The time that each component takes to come off the column is called its retention time and can be used to help identify it. Here, the HPLC instrument is being used to separate a mixture of two steroids used in a pharmaceutical preparation. The column chosen is packed with a nonpolar solid. The tails of the molecules represent hydrocarbon chains C8H17. Having chosen the solvents, detector wavelength and flow rate, a single sample is run by injecting about 20 microliters into the injection port. The more polar component comes off the column first, followed by the less polar. The peak at retention time, 1.5 minutes, represents other ingredients used in formulating the product. This is the pharmaceutical product, and behind it, its chromatogram.
Okay. Now comes fast performance liquid chromatography, which is a higher version of Hplc, where solvent velocity is controlled by the pumps, and it's controlled the flow rate of solvents. The solvents are assessed through the tubing from outside reservoir, and the flow rate of the solvent is set through the computer input and controlled by the pumps. Now comes the gel electrophoresis. So this is done for separating the most popular, like your gel, that is a polyacrylate gel, which forms a three-dimensional molecular sieve. So under denaturing condition, the gel can dissolve proteins of the size from 10 to 50 kilodalton, which is quite powerful. And proteins and nucleic acids migrate in an electric field according to their net charge. And for this reason, separation can be achieved in many cases. An electrophorese is coupled to a second separation method to achieve very high resolutions of different proteins. The most popular method is Sds Page, where electrophorese is coupled to a sieving device that differentially retards large proteins. So Sds Page, the key feature is that all Sds coated proteins have same electrophoretic mobility. So free flow electrophoresis, this is the simplest in concept but most difficult to achieve and requires expensive equipment. So basically, in this we have a protein mixture which is injected at the top of the plates, begin to flow with the buffer flow, and proteins that carry the negative charge will flow migrate towards the positive electrode, while the positive charge proteins go to the opposite direction. So buffer flow, if we see, buffer flow is adjusted so the proteins never reaches the electrodes before it arrives at the bottom of the plate, where different fractions are collected in small tubes. So at the bottom of the plates, a beam of UV light at 280 nanometer across the absorption due to the pain is absorbed. So this is the simplest case of gel electrophorese. You put our sample in the figure A, then it will migrate according to the sizes. Smallest will go at the bottom, the middle size will be in between, and the yellow will be at the top. And then in the gel electrophoresis, you have acrylonite and methyl bisacramide, and you make a polymers of them. And because of these polymers, your samples get collected in these pores. And also Sds Page is there. Sds is done in order to coat your protein. So it's a give a negative charge. It will open up your proteins from your primary structure, from a quaternary structure to primary structure. And based on that, again, because of the Sds Page, according to different sizes, you will separate your different bands. So in this way, in the Sds Page, we got these kinds of bands: smaller, 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 larger bands like this. And we can save these results for the later also, some different bands. And further, before you go for gel electrophoresis, there is called 2D gel electrophoresis, which is based on isoelectric focusing gel electrophoresis. So in this, basically, you are having a high pH and low pH, and you separate your protein particles according to the pH level. So the higher pH will be, they will find their isoelectric pH point, that is Pi value, and according to that Pi value, you will separate them. So once you separated them, you can put them through the 2D gel. This is the 1D gel at the moment. So let's check how you prepare your protein for precipitation for 2D gel. This will not have any sound. We have to see the text at the bottom. So this is how we do the protein. So class, we will do the rest two-dimensional electro forces in the evening class. So I can stop it here, and we see us in the evening then. Huh? So we see us in the evening at 7 p.m. I will write it there. Okay, class. Thank you.